This would be Module 1, Section 2. We're going to complete Chapter 3 and go on to Chapter 4 of our textbook. This is for the Anatomy and Physiology online class. And speaking about the cells in Section 1, we just review a little bit about the cell. We know that cytoplasm is the living material within the cell, and the nucleoplasm, of course, is the living material within the nucleus. All of it in times past was called protoplasm. And of course, the nucleus is the control center. It has the DNA. It may have a nucleolus, which would be an area where RNA is refined and RNA is also stored. The cell membrane is a single membrane. The fluid mosaic model is the one accepted now. And then the nuclear membrane is a double membrane. Other double membrane structures would include the endoplasmic reticulum and the Golgi body, and of course the mitochondria, which is the powerhouse. Single membrane structures would include the vacuoles. And then a lysosome is another single membrane structure. The lysosome is also called the digestive bag or suicide bag because it contains proteolytic enzymes, and if this membrane breaks open inside the cell, then the enzymes will break down the cell and destroy it. Other structures would include tubular structures such as the centriole. And the centriole is a short cylindrical assembly of microtubules arranged in 9 groups of 3 microtubules each. You need to refer to figure 3.33, I believe, in your textbook. And the 2 centrioles are usually perpendicular to each other within a small clear area of cytoplasm, which is called the centrosome. So the centrosome contains the centrioles. These are going to be involved in forming spindle fibers, which will be used in the processes of mitosis and meiosis, both discussed in Chapter 4. Inclusions also would be structures such as granules that are stored within the cytoplasm, and perhaps pigments or fat droplets. Sometimes foreign bodies such as viruses and bacteria might also be here. Another activity of the cell, though, is getting material into the cell and getting material out of the cell. Getting material into the cell is referred to as endocytosis, and one particular way is through pinocytosis. In this case, sometimes this is called cell drinking. And the materials land on the cell membrane. The membrane invaginates, which does require some energy. It loops back around the material to form a little vacuole, which is brought into the cell. There, materials can be combined with the lysosome, and which would digest the materials, and then what's left would be, and when that combines with the lysosome and the pinosome, it's called a pinolysosome. The enzymes that were in the lysosome break down the material brought in through the pinosome, and then digested materials move through the membrane out into the cytoplasm, and then what's left that's not digestible is called a residual body. Okay, now the other process would be evagination, known as phagocytosis or cell eating, and part of the membrane would move out and around, perhaps forming pseudopods or false-like arms that surround material, then bring it in, forming a vacuole called a phagosome. The phagosome combines with the lysosome to form a phagolysosome. Digestion occurs, materials move out into the cytoplasm that were digested, and then anything that's not digestible, whatever remains, is this together is called the residual body. One of the theories of aging is that the cells fill up with residual bodies, and that causes, of course, perhaps a deterioration of the cell. Sometimes the residual bodies are pushed to the outside and by exclusion. Other times they stay within the cell. But those 2 examples of the bringing materials into the cell. Into the cell would be endocytosis. Outside would be exocytosis. The Golgi body packages materials that are produced and then sends them, forms membrane around them, and then they move to the cell membrane and combine with it to be released, and then contents of that, the vacuole is released to the outside of the cell. That would be exocytosis. As we move on into chapter 4, we see again about the DNA and the RNA, and then the processes of semi-conservative replication, making DNA, duplicating, and then the transcription, using DNA to form messenger RNA, and then translation using the messenger RNA along with transfer RNA, which will link to amino acids, and then the transfer RNA comes in and connects with the messenger RNA. The 3 nitrogenous bases in a row on the messenger RNA are called a codon, and they code for a specific amino acid. And there are many of those codons to make the protein. But on the transfer RNA, it looks sort of like a little clover leaf with one side holding the amino acids, and then a rounded part of the clover-like leaf holding 3 nitrogenous bases. Those 3 bases are called the anticodon, and the anticodon then will match with the proper codon on the messenger RNA at the ribosome. Then enzymes will link the amino acids that the transfer RNA brings in one at a time, and they link those together to form the protein. It takes at least 50 amino acids to make a small protein, and that means it would be 50 times 3 is 150 nitrogenous bases would be involved. Okay, but then that would form the protein synthesis, which is an anabolic process. Anabolism is the building up of materials. Catabolism is breaking that down, and the catabolism would be such things as the glycolysis, the electron transport system, and the Krebs cycle. The electron transport system and Krebs cycle, of course, occur in the mitochondria. Glycolysis occurs in the cytoplasm. You have bigger molecules broken down with energy released to form ATP, and then in anabolism, building up, you require ATP to link materials together, so energy is required. But those are the 2 phases of metabolism that occurs in the cell. As we continue on, though, in chapter 4, the 2 processes that are going to be very important to know would be mitosis and meiosis. With mitosis, this will be somatic cell growth, and meiosis is called reduction division to form the gametes, or reproductive cells. Well, with mitosis, there's several phases. The interphase, we call it resting. We put resting in quotes because it's only resting from the actual division. In the interphase stage, it's usually the longest stage, and interphase has 3 parts. It has a G1, an S, and a G2. G1 materials are being pulled together that are going to be needed to make more parts. In S stage is synthesis. That's when more DNA is produced and increased. And then in the G2, that's when it's preparing for the actual division. There's more detail than this that you need to study in your textbook. But once it's gone through the interphase stage, then the next stage would be prophase. And in prophase, the chromatin material starts to shorten and thicken to form the chromosomes. And then they begin to gather. The cell membrane breaks down, and nucleolus disappears for a period of time. Then at metaphase, the chromosomes line up, the double chromosomes line up just randomly at the equator, in about the middle of the cell. The centrioles produce spindle fibers, and they attach to the chromosomes, one for each pair. The centromere holding the duplicated chromosomes together breaks down, and then it moves on into anaphase. And that's when the spindle fibers are pulling this apart or pushing to the other side, probably pulling it apart. And so once they start breaking away, then we say that this is anaphase. Continues pulling on toward the spindle fiber. The spindle fibers keep pulling until the centromere that's at each side. And then finally, it starts breaking apart into 2 cells. If it's a plant cell, there'll be a plant cell wall produced across the center, a plate. And then if it's an animal cell, it just pinches in. But once you have the 2 cells, then that's called telophase or telophase, both having the 2N or diploid number of chromosomes looking just like the parent cell. And this is how our growth occurs. As we see here, we see trees in this picture. People are fishing, and so the fish grow, the trees grow, the grass grows. We grow, everything, living things primarily grow by the process of mitosis. Again, that is interphase, prophase, metaphase, anaphase, and telophase. The other process is meiosis, which is reduction division, and that only occurs in the formation of the gametes. That would be in spermatogenesis for the male gamete, sperm, or spermatozoa, and oogenesis for the formation of the egg. Okay, in both cases, the chromosomes duplicate, and then but there's a little bit of difference. When they line up at the equator, they're going to line up in tetrads. The two, say for chromosome 1, you have one from the mother, one from the father, one male, one female. They've doubled, and now they line up together, not randomly. So the like chromosomes line up in those duplicated pairs across the equator. The spindle fiber attaches. The centromere does not divide at this time. Spindle fibers attach, and when they start pulling apart to form anaphase 1, it's going to be one pair or the other going to opposite sides, not one of each pair. And then when that forms and pinches apart, then you're going to have them lining up again in metaphase 2 at the equator. Spindle fibers attach. Now the centromere divides, and as it pulls apart, it's one of each pair present going to opposite poles. And so there are two divisions in meiosis, and with the end result being 4 cells, each having the N or haploid number of chromosomes. In spermatogenesis, there's an equal cytokinesis or division of the cytoplasm. And so that there are 4 equal-sized cells that are formed, each having the N or haploid number of chromosomes. In oogenesis, then with the first metaphase, they've lined up the same way, duplicate pairs from male and female, as tetrads lining up, and then again, one pair or the other going to opposite poles. But when the cytoplasm divides, there's an unequal division of the cytoplasm, so that there's one large egg cell and one polar body. Okay, now it lines up again, the chromosomes line up again at the equator, and at this point, the centromere divides. And again, there's a pulling apart for the anaphase 2, but with the polar body divided into two smaller polar bodies, and the egg dividing into one egg and another polar body. So there will be 4 cells formed, but there are 3 very small polar bodies that just carry the nuclear material, the N number of chromosomes, and then one ovum or egg that's capable of being fertilized. The egg is going to contain nutrients. It contains a lot of cytoplasm. It contains the organelles that's going to be needed for the possible embryo that's formed. But there will be an unequal division in the oogenesis. There's an equal division in spermatogenesis, but an unequal division of cytoplasm in oogenesis. Now, at the end of oogenesis, then there are 3 small polar bodies and 1 egg or ovum, and only the egg or ovum is capable of being fertilized. If fertilization occurs in the sperm combined with the egg, then that's N plus N, or 2N, so the 2N number of chromosomes, the diploid number, is reestablished, and that would be a zygote, a 1-cell structure, and from that point on, growth is by mitosis, interphase, prophase, metaphase, anaphase, telophase, and it continues on. So this will be the conclusion of Module 1, Section 2, going through Chapter 4.